Hello, welcome back to session 6 of Unit 2. And under this session, we'll be looking at the second part of the movement of substances in and out of cell. At the end of this session, you should be able to explain active transport, distinguish between cold transport and bulk transport, and state three examples of endocytosis, and also name the process by which white blood cells protect the body from microorganisms, and finally describe how hypercholesterolemia develops. For active transport, there is movement of solute through a membrane against concentration gradient that requires the use of energy in the form of ATP. Some transport proteins, however, move solutes against their concentration gradient. The solutes move across the plasma membrane from the side or region where they are less concentrated to the region where they are more concentrated. And this type of membrane transport in which energy is used or requires the use of energy in the form of ATP is referred to as active transport. The active transport make use of carrier proteins other than channel proteins. So, and a typical example is the sodium-potassium pump transport system. And this system requires the use of energy from ATP, whereby the terminal phosphate group of ATP is transferred to the carrier protein. So, in the cell, the sodium ion concentration is much more in the cell cytoplasm as compared to the extracellular fluid. So ATP donates its terminal potassium as demonstrated in figure 11. So as the phosphate group is attached to the transport protein, it results in the change in shape of the carrier protein that will enable to transport the solute across the membrane. In this case, three sodium ions will then be transported across the plasma membrane to the extracellular fluid, which results in the pumping of two ions of potassium rather into the cell. Another mechanism of the transport of solutes or substances across the plasma membrane is co-transport. And with regard to co-transport, a single ATP powered pump that transports a specific solute results in indirect drawing of other solutes into the cell, and this process is co-transport. During plants, cells that make use of the gradient of hydrogen ions to drive the active transport of amino acids, sugars, and many other nutrients into the cell. And as a result, it leads to diffusion of hydrogen ions back into the cell through one transport protein coupled with the transport of sucrose into the cell, and this occurs against concentration gradient. These processes has helped in the effective treatment of dehydration resulting from diarrhea in humans, and this application of co-transport, especially in patients with diarrhea, whereby they are given a solution containing high concentration of glucose and salt to drink, and at the end, the solutes are taken up by transport proteins on the surface of the intestinal cells and are therefore passed through the cells into the blood. So the rise in this blood solute concentration results in the flow of water by osmosis from the intestine through the intestinal cells into the blood. So far, the processes we have discussed are meant to transport water and other small solutes into the cell across the plasma membrane. What of other bulky substances such as proteins and polysaccharides? So now we'll have a look at bulk transport. For bulk transport, it involves the movement of large molecules, which includes proteins and polysaccharides, across the plasma membrane in bulk, and this mechanism involves the packaging of these large molecules into vesicles. Bulk transport is also a form of active transport in that the process requires the use of energy in the form of ATP. There are two main types of bulk transport, and these are exocytosis and endocytosis. We will have a look at these two main types in detail in the subsequent slides. So beginning with exocytosis and our bulk transport. Exocytosis involves the expulsion of certain biological molecules from the cell by the fusion of vesicles with the plasma membrane. And the transport vesicle from the Golgi apparatus will travel along microtubules of the cytoskeleton and therefore joins onto the plasma membrane. So the two bilipid layers rearrange themselves so that the two membranes will fuse. The content of the vesicle therefore spills to the outside of the cell, but the membrane becomes part of the plasma membrane. For instance, the pancreas produces insulin and secretes it into blood by exocytosis. The nerve cell also uses exocytosis to discharge neurotransmitters that signal other neurons or muscle cells. And then also in animals, cells which produce and secrete digestive enzymes do so by exocytosis. That's when a plant cell also are making their walls. Exocytosis helps in delivering protein and carbohydrates from the Golgi vesicles, which are used to build the cell wall. Now let's have a look at endocytosis as a type of bulk transport. Endocytosis is basically the uptake of substances out of the cell into the cell. So in endocytosis, the cell membrane forms vesicles from biological molecules and particulate matter from the surroundings, and the small area of the membrane sinks inward to form a pocket, and this eventually develops into a vesicle trapping the material which was initially outside the cell. There are three main types of endocytosis, and the first one we'll be looking at is phagocytosis. Phagocytosis literally means cell eating, and in phagocytosis, the plasma membrane invaginates to form a pseudopodia that encloses the particle. The invagination therefore becomes sealed off as a phagocytic vesicle or food vacuole particle. The ingested particle becomes digested when the vacuole fuses with a lysosome that contains hydrolytic enzymes. The end product of digestion is then absorbed into the surrounding cytoplasm. For instance, the white blood cells protect the body from microorganisms through phagocytosis. Also, amoeba feeds on small organisms by phagocytosis. The second type of endocytosis is pinocytosis, and it literally means cell drinking. In pinocytosis, droplets of extracellular fluids are carried into the cell by tiny vesicles, and it is not the fluid that is needed by the cell, but the molecules dissolved in it. And so, owing to the fact that any solute can be taken into the cell, pinocytosis is non-specific in the substances it transports. So enzymes are then discharged from the surrounding lysosomes to break and release the molecules. So for instance, in humans, maturing human egg cells takes up nutrients from their surrounding cells by pinocytosis process. Now, the third type of endocytosis is receptor-mediated endocytosis. And in receptor-mediated endocytosis, there's uptake of bulk quantities of specific substances by cells, and this may not be necessarily abundant in the extracellular fluid. There are specific receptor proteins that are located at certain regions of the plasma membrane, and they are found on the cytoplasmic side of the plasma membrane. So these regions are called coated plates, and these coated plates binds to ligands, or ligands binds to these protein receptors. The coated plates form vesicles containing the ligand molecules. So the ingested material then is liberated from the vesicle and they are recycled to the plasma membrane by the same vesicle. Figure 12 indicates the types of endocytosis, that is phagocytosis, pinocytosis, and receptor-mediated endocytosis, as already explained in the previous slides. Now, human cells make use of receptor-mediated endocytosis to take in cholesterol that is used for the synthesis of membrane and other steroids. And cholesterol travels in the blood as low-density lipoproteins. These lipoproteins or low-density lipoproteins binds to the low-density lipoprotein receptor sites on the plasma membrane and enters the cell by endocytosis. So in humans, when there is defective or absence of low-density lipoprotein receptor proteins, there's buildup of lipid deposits within the walls of blood vessels. So the lumen of the blood vessels become narrow and when the blood flow is impeded. So this results in the condition referred to as hypercholesterolemia, which is an inherited disease in humans and is characterized by a very high level of cholesterol in the blood as a result of or resulting in atherosclerosis. In summary, under this session, we've learned about active transport, which involves the movement of solutes across the cell membrane against concentration gradients, requiring the use of energy in the form of ATP. And we've also learned the movement of amino acids and sugars occur by co-transport, while for proteins and polysaccharides or large molecules do so by bulk transport. And there are two types of bulk transport, being exocytosis and endocytosis. And we looked into detail of each of the type of the bulk transport. And we also finally looked at hypercholesterolemia, which results when there is absence or defective low-density lipoprotein receptor proteins that could lead in the buildup of cholesterol in the blood vessel.